Assalamualaikum warahmatullahi wabarakatuh dan salam sejahtera bagi kita semua. Atas nama Sivitas Akademika dan seluruh warga Fakultas Sains dan Matematika, saya Profesor Dr. Widowati, Dekan Fakultas Sains dan Matematika Universitas Diponegoro, mengucapkan selamat atas diterimanya saudara-saudari di Fakultas Sains dan Matematika.
Assalamualaikum warahmatullahi wabarakatuh. Program Studi Bioteknologi Universitas Diponegoro berdiri pada tahun 2018. Program Studi Bioteknologi memiliki visi pada tahun 2025 menjadi pusat pendidikan, penelitian bidang bioteknologi sumber daya hayati yang berkontribusi dalam skala nasional dan internasional dalam pemanfaatan pengembangan sumber daya hayati yang berkelanjutan. Untuk selanjutnya, misi dari program studi bioteknologi Universitas Diponegoro terdiri dari 1. Melaksanakan pendidikan tinggi jenjang sarjana bidang bioteknologi sumber daya hayati yang unggul, bermoral, beretika, berkompeten, berkarakter, dan berwawasan kebangsaan. Misi yang kedua adalah melaksanakan dan meningkatkan kualitas penelitian yang kreatif dan inovatif dalam pemanfaatan pengembangan sumber daya hayati yang berkelanjutan berdasarkan prinsip-prinsip bioteknologi. Misi yang ketiga adalah melaksanakan dan meningkatkan kualitas dan kuantitas pengabdian kepada masyarakat melalui pendidikan tentang pemanfaatan dan pengembangan sumber daya hayati yang berkelanjutan.
Okay, good morning, everyone. Good morning, Ajahn Op.
Yes, good morning, Ajahn Anto. Nice to meet you.
Nice to meet you again.
Yes,
It's a long time we don't see each other.
yes.
Okay. Good morning, everyone. Welcome to visiting professor program at Biotechnology Diponegoro University. And today it's my honor to introduce to you our speaker in this visiting program. Before we start our guest lecture, I would like to give you a little bit curriculum vitae of our speaker today, Professor Panlada Titabut, PhD. Okay, Professor Titabut has a background in PhD in biotechnology from School of Biotechnology, Suranare University of Technology in Thailand. And at the moment, she is one of the professor in School of Biotechnology, Institute of Agricultural Technology, SUT. And, wait a second. Her research interest is nitrogen fixation in bacteria, inoculation and biofertilizer production and development, plant microbe interaction, plant growth promoting rhizobacteria, and microbial degradation of environmental pollutants. She has more than 100 publication, international publication. So I think she's very expert in her field. And to let you know that this visiting program is held by Biotechnology Study Program. Our aim is to give, to enrich the knowledge and also to give insight to the student as well as a participant in this program. And the student is not only coming from biotechnology, but also from biotechnology and from not only from Diponegoro University, but there are many from other university. So we would like to share your knowledge and excitement on this program. So to make fast, I think I will give time for you, Ajahn Op, please. Time is yours. Wait a second, you are still in mute. Please, host, can you unmute?
Yes, okay.
Okay.
Can everyone hear me now?
Yes.
Yes, okay. Thank you, Ajahn Anto. So I am so excited today to talk with all students. Actually, there is more than 100 students now. Normally my class is maybe only 30 or 40 students. So this is my first time to teach like more than 100. But anyway, my talk or my English may be not so good, but if you have any question, you can interrupt anytime. Okay, you just open your microphone and then just ask the question. Okay, for all students, no problem. Okay, and can I share the slide in the chat? Is it okay? So everyone can get the PDF file and maybe they can take some note. Okay, so I will share the slide. Please. Okay. Let's see. So I'm sharing it now, so it's quite big, so please wait for a while.
Yes.
Okay. So I hope every student can download and you maybe can take some note on that PDF file if you like. And meanwhile, may I share the slide, Professor Anton?
Yes, please.
Okay, thank you. Share. So can everyone see my slide now?
Yes.
Yes. Okay. So I make it full like this.
Okay, cool.
Cool. Okay, thank you. So that student, every student can get my slide from the chat. And any
Okay,
problem?
yes.
Yes. Okay. Good. All right. So I will start from this topic because I think this is the topic that I have a lot of research on this. But actually, it's not only myself. I also work with Professor Ning Amrung, Professor Nantakon, and also other students in the lab. Okay, so they work very hard to do the research on biofertilizer, on the plant growth promotion, and on the plant and microbe interaction. So today I'm not sure if all students have the basic knowledge on plant microbe interactions or not. So today I will try to put some basic knowledge and also to give some information about the laboratory of my research and how to apply it in the field. Okay, so that's why this is the topic in today is the plant microbe interactions application of PGPR in Thailand agricultural industry from lab to the field. Okay, but I would like to say that actually in Thailand, the industry about the PGPR or biofertilization, biofertilizer production is not so many, it's not so big, but I will try to explain you why. Okay, all right, let's start. Okay, so all students, first of all, I would like to introduce you about the microorganism. I hope everyone know microorganism, right? What is microbe or microorganism? So microbes, it means any living organism that's very, very small, right? It can be bacteria, fungi, right? Or protozoa that we cannot see by naked eye. So we can call it microorganism, okay? But when we talk about the interaction with plant, okay, when we talk about the interaction between microbe and plant, there are two types of microbes. First, it can be the enemy, okay, and the other one, the microbe, can be a friend. Okay, so when we talk about the enemy, it means that those microbes can be a pathogen, right? The pathogen can cause plant diseases. Okay, so this is the pathogen, this is the microbe as the enemy, bad microbe. But today we are going to talk about the good microbes, right? Microbe as a friend. So we can apply this microbe for crop production or for plant growth promotion. Okay, in the topic, I give you the word PGPR. Okay, I give you the word PGPR, but what is it? What is it? Okay. So in terms of PGPR, I will tell you about the meaning again, but actually the PGPR is the bacteria, right? The P is come from the plant, G is mean growth, P is mean promoting, and R is rhizobacteria. Okay, so it means that this PGPR is the bacteria that stay around the rhizosphere, around the root. Okay, and these bacteria have the function to promote plant growth. But to promote plant growth, it can be promoted by serving the nutrient, okay, or promote by indirect way. For example, can be a biocontrol, right, to control the plant pathogen, to control the plant disease. Or the direct way is the promote the plant growth by provide the nutrient, for example, provide the nitrogen, provide the phosphate, okay, or some phytohormone, or some iron, okay. But for biocontrol, this is the indirect way. They can promote plant growth by degrade the cell lysis of pathogen of the fungi, or produce some antibiotics to inhibit the growth of pathogen. Okay, so when we talk about the PGPR we talking about the good bacteria, right? The friends of plant, the good bacteria that can promote plant growth in direct way or in indirect way, biocontrol. Okay, so this one benefit to plant growth. Okay, so we are talking about the good microbes or good bacteria. Okay, so if you have any question, please just open your microphone and ask anyway. No, there is no problem. Okay, so this is the meaning, plant growth, right? Promoting rhizobacteria, so PGPR. Okay, this is the meaning is very easy. Just promote the growth of the plant. But I would like you to mention first thing is the rhizobacteria. Why we have to talk about rhizobacteria? Okay, so you know what is rhizo? Rhizo is the root. It means root, right? Plant root. And bacteria is bacteria, right? So when we're talking about PGPR, we're talking only the bacteria that stay, right? Stay around the rhizosphere, around the root. Okay, so if you see this picture, when you dig up the plant, okay, you will get some soil, you will get the root like this. So the bacteria that stay around the root in the soil like this, okay, it can be stay around the root like this in the soil. If you see, this is the soil, right? It's in the brown color. The bacteria that stay around the soil or stay on the rhizoplane. The rhizoplane is mean the surface of the root, right? You can see the rhizoplane. The rhizoplane is mean that the bacteria that stay nearby the root in the soil or attached on the surface of the root. Okay, surface of the root. We can call it rhizobacteria. Okay, and some definition, some scientists also, they say that the rhizobacteria can also involve in the bacteria that stay in the root cortex. Okay, so if you see, this is the vascular tissue, right? And this is the root cortex. So some bacteria that stay in the root cortex, we can call endorhizosphere, right? This endorhizosphere, this ectorhizosphere. So when we talk about rhizobacteria, it can be the bacteria that stay in the soil nearby the root. It can be the bacteria that attach on the surface of the root. This is we call rhizoplane. Or the bacteria that stay inside the root cortex. Okay, so these bacteria we call PGPR or rhizobacteria that can promote plant growth promotion. Okay, so I would like you to know this definition first before we go further. Okay. So if you have a question, please open your microphone and ask. Okay. All right. So now you know the definition of PGPR, but actually the PGPR, there are so many types. Okay, there are so many types. For example, the first type we call is symbiosis. Okay, symbiosis, right? So when we're talking about the bacteria that stay nearby the root or on the root surface or inside the root cortex, some bacteria can symbiosis with plant. So this one, this is the close or prolonged association between plant and bacteria. They can exchange the benefit. For example, rhizobium. Okay, I will have some example later to show you. Okay, so this rhizobium, they can make the nodule on the plant root, especially with the leguminous plant, okay, like soybean, mung bean, peanut. So bacteria can make the nodule and they can symbiosis by exchange between the nitrogen fixation from bacteria to provide with the nitrogen fertilizer. In the same time, the plant have the photosynthesis and then provide with the sugar or carbon source to bacteria. Okay, this is the first type of PGPR. So it means that this type of bacteria can promote plant growth also. The second type is root associative. Okay, root associative bacteria. This type is normally meaning about the bacteria that stay just nearby the root or just attached on the root surface without going inside the root tissue. Right? They just associate, they just attach on the root surface or stay nearby. So this one we call free living bacteria and just colonize there in the rhizoplane. Okay, and the last type of PGPR we call endophytic bacteria. This one is similar to the symbiosis, because symbiosis, they stay inside the cell, right? But for symbiosis, they will have the positive interaction, right? The plant gives the sugar, the bacteria gives the nitrogen. But for endophytic bacteria, this one is mean that the bacteria stay inside plant tissue, but it's not necessary that they have to provide the plant with benefit, or it's not necessary that the plant gives the bacteria with the food or with the sugar. When we term endophyte or endophytic bacteria, just meaning any bacteria that stay inside the plant tissue. Okay, but it is not necessary that the plant will give the benefit to bacteria or not, but just the bacteria stay inside. But when we're talking about PGPR, right, because this is a plant growth promoting rhizobacteria, the endophytic bacteria that we are going to talk in this class, this bacteria will provide the benefit to plant and stay inside the plant tissue. And it not stay only in the rhizosphere, not in the root. It can stay in the stem, it can stay in the leaf, it can stay in the flower, right? So it just can go to every part of the plant tissue, okay? So this is 3 types of PGPR. And I will tell you more detail in each type of PGPR and also about the application of this bacteria, okay? All right, okay, so the first one is the symbiosis, okay? For the symbiosis, so you can see that this is the nodule, okay? This is nodule. So the line mainly is rhizobium, as I told you. So the rhizobium can make nodule on the leguminous plant like this, and you will see that the effective nodule, if you cut the nodule, you will see the red color inside. But once you cut some nodule and you see the white color or brown color or green color, it means that the bacteria inside or rhizobium inside is not efficient, right? The good bacteria or the good rhizobium, when you cut the nodule, today you can go back to your home if you have some leguminous plant, take the plant, right? Take the root and see if you see the nodule, cut it half. If you see the red color like this, it means that your rhizobium is very high efficiency for nitrogen fixation. So if you use the spatial microscope, we call confocal microscope, you will see the small dot. You can see small dot like this. This is the cell of rhizobium inside the plant. This is the plant cell, right? And you will see a small rhizobium cell, the bacteria inside the plant cell like this. And this bacteria can fix nitrogen and then provide the nitrogen to plant. Okay. In the same time, the plant do the photosynthesis and provide the sugar or the carbon source for bacteria for growth. Okay, so that's why we call symbiosis, because both of them give the benefit to each other. Okay, and another type is root associative, right? As I told you, this one they just stay near the root, right, inside the soil, right, or attached on the root surface, right? So this one you can see the different type, right? This one is stay with the symbiosis. They make the special nodule like this. But for the root associative, this bacteria just locate on the root surface or in the soil, right? But it does not go inside the plant tissue. Okay, this is a root associative. Most of bacteria in PGPR follow in this type. Okay. And another one is endophytic, as I told you. Endophytic bacteria, right? This bacteria can stay inside the plant tissue. They maybe can go to the crack entry or somehow. We don't know the mechanism yet how the bacteria go inside the root or the cell, the plant cell. But these bacteria, when they go inside the plant cell, they can stay in the root like this, okay? And it can also, once they go to the root tissue, they can go up to the stem, or they can go up to the leaf. So you can see that the bacteria can stay in the leaf tissue like this. So it means that when we talk about endophytic bacteria, It means they stay inside the plant tissue, in root, in stem, flower, in leaf. Okay, but when we talk about PGPR for the endophytic bacteria, this bacteria will benefit for plant growth. Okay, all right. So let's start with the plant microbe interaction. What we do in the lab and what we study in the lab. Okay, and how we take the knowledge from the lab and apply in the field. Okay, so I will start first with the first type. Okay, the symbiosis group. Okay, the symbiosis group. So in the symbiosis group, as I mentioned, mainly we focus about the Lysobium. Okay, so Lysobium is the name of the bacteria. Okay, this is the name of bacteria. This is one genus. Actually, Lysobium have several genus. For example, Rhizobium, Bradylysobium, or Mesolysobium, Azolysobium. This is just the name. Okay, the name of the genus. But the common name of this type of bacteria we call Lysobium. I think you may know a lot about Lysobium because in biology, many professors always show the example of symbiosis using Lysobium, right? The Lysobium can make the nodule, right, as I show you. And then this Rhizobium can fix nitrogen from the air, right? And then also provide the nitrogen in form of ammonia to the plant. Okay, so the limitation of using Lysobium is the specificity. What does it mean? It means that Lysobium can use only with leguminous plant, right? We cannot use Lysobium. Lysobium cannot make nodule with rice. Lysobium cannot make nodule with sugarcane or other plant or any vegetable. Okay, but Lysobium can make nodule and symbiosis with only leguminous plant. So that's why this is the limitation. We know that Lysobium can fix a lot of nitrogen, right? They can uptake the nitrogen from the air, right? Because in the air we have a lot of nitrogen, about 72%, right? So the nitrogen in the air, the Lysobium can take and then provide nitrogen to the plant. But the limitation is we can use only with leguminous plant. And more specific is some Lysobium can use only with specific legume. For example, if I use the Bradylysobium can use only with mung bean. This Lysobium can use with peanut. But some Bradylysobium, if we use with mung bean, sometimes cannot use with soybean. It's very specific, right? Or the Bradylysobium that nodulate Cotarrhea. Sometimes we cannot use it for nodulate peanut. Okay, so very specific. So when you select the Lysobium to use with legumes, you have to select the specific one, right? You have to select the specific one. Lysobium for soybean, Lysobium for peanut, or Lysobium for mung bean. Okay, this is very specific. This is the limitation that we cannot use Lysobium with other plants. We cannot use it to make nodule or fix nitrogen like this. Okay, so the student may ask why this Lysobium is so specific, right? Why it's so specific? Why this Lysobium specific to soybean? Why this Lysobium specific to mung bean, for example, or to peanut, right? So in our lab, we study about the nodulation mechanisms. What does it mean, nodulation? Nodulation is come from nodule. Okay, when we talk about nodule is mean this one, nodule. This is we call nodule, right? The organ like this we call nodule. Okay, so the word nodulation is mean the process that bacteria, right? The bacteria go inside the plant cells and making the nodule, right? Making the nodule we call nodulation. Okay, So why this modulation or this mechanism are so specific, right? Why it's so very specific? Because in this symbiosis pathway, right, the plant and the rhizobium have some interaction. They have to talk to each other. They have to talk to each other. For example, this is the plant, right? This is the plant root. Okay, this is the plant root. In the plant root, they have to talk with rhizobium, right? This is the rhizobium bacteria. They have to talk to each other. How they talk? Okay, how they talk? They talk through the mechanism called not factor dependent. The student may have question, what is not factor? Okay, this is not factor. This is a chemical compound produced by rhizobium. Produced by rhizobium. This is not factor. This is a chemical compound produced by rhizobium. Okay, why we call not factor dependent and why how they talk? Okay, they start talking from first. The legume will secrete or will produce some chemical compound we call flavonoid. Okay, this flavonoid is very specific. Soybean will produce one type of flavonoid. Peanut will produce another type of flavonoid, right? And mung bean also produce another type of flavonoid. It means that different plant, different legume produce different types of flavonoid. Okay, this is the first conversation between plant and microbe. So the first speaker is the plant. They talk by secrete the flavonoid. And then after that, the bacteria or the rhizobium, they will receive this signaling, right? They will receive this signaling. And if this signaling is correct, for example, if this is the rhizobium for peanut, for example, and the peanuts produce the green compound, the green flavonoid. And if the rhizobium can receive the correct green compound, then the rhizobium get the signal and produce the NOT factors. Okay, the rhizobium will the NOT factor when, right? When only they get the correct signaling from plant. Okay, what that is it means that if this rhizobium is for soybean and used with peanut, the peanut produce the green color of flavonoid. It means that if this rhizobium is for soybean, this rhizobium cannot get the signal from peanut. So they cannot produce the NOT factor. Okay, so can everyone follow me? Right? It means that the first speaker is the legumes. The legume must produce some flavonoid, the signaling. And different legume produce different signaling compound, right? And if this signal is correct and use the correct rhizobium, this signal can induce the rhizobium to produce NOT factor. Okay, and this NOT factor will secrete and then go to the plant. Okay, go to the plant. And once the plant receives this NOT factor, this is like a key. Okay, this is like a key. This key will open the signaling in the plant. Okay, there are so many steps in the plant that I cannot explain you now because it takes very, very long explanation. Okay, but you know that there are so many steps in the plant signaling after they got the signal from bacteria, after they got NOT factor from bacteria. And the plant will check, again, the plant will check if this NOT factor is correct. Okay, then the plant will start the signaling to produce the nodule, right? They will start the signaling inside the plant and produce the nodule. So it means that the bacteria can go inside and then making the nodule. Okay, this is why we call NOT factor dependent, because they need the specificity from plant. They need the NOT factor very specific. If this NOT factor cannot bind with plant, the plant cannot have this signaling. So they have like two layers of signaling. First from the plant and the second from the bacteria. Okay, they have two keys, right? Two keys. So that's why we call NOT factor dependence. Okay, and if you use another type of bacteria, if you use another rhizobium that's not specific, this NOT factor cannot bind or cannot induce the plant and to make the nodule. Okay, so this is why we call not factor dependent. Okay, and another mechanism that we found recently, right? We found that some lysobium, they have the mechanism similar to pathogen, right? The pathogen does not have the gene to produce NOT factor to be a key to unlock this signaling. The pathogen have another system, right? Another mechanism to go inside the plant. And we found that lysobium also have this system to go inside the lysobium. So that's why we call NOT factor independent system, right? This one is NOT factor dependent, but this one is NOT factor independent, right? This factor we use the system called the secretion system. What is secretion system? Okay, secretion system is the system that the bacteria produce the protein, right? Produce the protein. And this protein is like a syringe, you know, the syringe for injection, right? The syringe. This syringe produced from bacteria, and this syringe can go to the plant cell. So this is the plant cell, right? This is a plant cell. This is the bacterial cell. But this protein is like a syringe for injection. They put this syringe inside the plant cells, and then they produce the protein called effector protein. This is the effector protein, go inside the plant cell. And this effector protein have the influence or have the roles on activation of nodule formation. Okay, but some effector not just only activate the nodule formation, some effector can inhibit the nodule formation too. I will explain more what is it. Okay, but now you have to know there are at least two mechanisms. First, use the NOT factor, but the other one use the secretion system. But these two systems are very important to make the specificity between lysobium and legumes to make a nodule and symbiosis. Okay, so this is the secretion system that I told you. Okay, in our lab, we do a lot of research in this secretion system, and also Dr. Anto's wife also study about this secretion system too. Okay, so I will tell you like this. The secretion system, as I told you, this is the system, the protein, right, that the bacteria produce. If you see, this is the bacteria cell. This is the outer membrane, and this is the extracellular, like a LPS, right? LPS, lipopolysaccharide. This is the like polysaccharide on the the polysaccharide on the bacterial cells. Okay, so the protein, this protein is like a syringe, as I told you. They make the protein and then put inside the plant cell, the green area. This is the plant cell. Okay, this is the host target or the plant cell. So it means that once the bacteria attach on the root surface, okay, the bacteria can produce the protein like a syringe and then inject inside the plant cell. And they can produce many protein called effector and then pass through the syringe, and this effector protein can go into the plant cell. So from bacteria, transfer to plant cell. Okay, there are many types of secretion system, right? Type 1, type 2, until type 6. Okay, but in our lab, the most effective one is type 3, right? Type 3 secretion system and type 4 secretion system. Okay, so there are two types that we mainly focus for nodulation. Okay, so in this picture, I show you the both mechanism. Okay, this is the plant cell. Okay, this is the plant cell. This is the lysobium. This is the lysobium. Okay, so as I told you, the first mechanism, they use the NOT factor dependent, right? So it starts from the plant host flavonoid, right? Produce the flavonoid compound from the legumes. And if this is correct signal, the bacteria or lysobium will produce the NOT factor. Okay, and the NOT factor will go to the plant receptor, and the plant receptor will check, okay, this is the good or the correct factor, then this one will induce the signaling in plant to make nodule, okay, to make nodule. But the other thing, the other pathway is the type 3 secretion system, okay. In this case, the bacteria will produce the effector. As I told you, they can produce the effector, and they can produce the silage, like this, the silage protein. And this silage protein can inject into the plant cell, okay? And then this effector protein will transfer from bacteria and go inside the plant cell. And as I told you, there are two types of effector. The positive effector is mean that this effector can somehow activate the plant signaling and make the nodulation. Okay, this effector is a positive effector. But some rhizobium can also produce negative effector. And this negative effector will induce the plant immune. And once the plant immune is induced, it will stop, right? It will stop the nodulation because it's produced the defense reaction. Okay, it will stop the nodulation. So it means that this rhizobium is depend on the mechanism. If they produce nod factor, they can make nodule. But if they have some bad effector, negative effector, even they have nod factor, if they have the bad effector, it can also induce the defense reaction or immunity in plant and inhibit the nodulation. Okay, so this is that why the mechanism of rhizobium to nodulate the legume is so specific, and we can improve it. Okay, in our research, we try to improve it. This is the example in our research, the role of type 3 secretion system. Okay, so you see that we test the bradyrhizobium ORS. This is just the name of the strain of the rhizobium with 3 different mung bean. Okay, this is the Vigna anquiculata, Vigna mango, and Vigna radiata, different type of bean, of mung bean. So you see that when we inoculated with the wild type, right, with the normal rhizobium, this one can fix nitrogen, right? You can see it can produce the green color on the leaf, right? But with the NI, it's been non-inoculation. It means we did not put any rhizobium. So we grow this one in the plant, that in the plant nutrient that have all the nutrients but without nitrogen, right? That's why when we did not inoculate any rhizobium, the plant cannot fix nitrogen and the plant produce the the yellow leaf. But when we inoculate with the rhizobium wild type, okay, they can make nodule and fix nitrogen. But when we make a mutation on the type 3 secretion system, it means that this one make the mutation on this one. It means that the rhizobium cannot send or cannot transfer the effector to plant, right? Because we make the mutation on this silage, right? What happened? When we mutation the type 3 secretion system, it seems like the rhizobium cannot fix nitrogen very well. It seems like less nodule. It means that the nitrogen fixation or the nodulation or the symbiosis require the type 3 secretion system. And this is the positive interaction, right? Because they require some positive effector to promote the nodulation, right? Once when we mutation, right? This is the symbol of deletion, right? The symbol of deletion. When we make mutation, there is no effector, no secretion system. The plant growth is less compared to wild type. Okay, same as Vigna mango, right? This is the same thing. When we make the mutation or when we make the secretion system not function, okay, this plant is very weak, right? Weak symbiosis. It means that the type 3 secretion system for this strain of rhizobium with these two plants are positive, right? It is a positive interaction. They need the type 3 secretion system. They need the positive effector. But in the opposite way, when we test with Vigna radiata, another species of mung bean, this rhizobium wild type, they cannot make nodule. they cannot symbiosis. But once we delete the secretion system, when delete the secretion system, this one can make nodule and produce the nitrogen fixation. What that is mean? It means that in this lysobium, they also have the negative effector, right? That's why when it still have the secretion system, right, the effector will stimulate the plant defense and inhibit the nodulation in wild type. But when we make the mutation on secretion system, there is no effector to produce. It means that this bad effector cannot go inside the plant, and only nod factor can induce, right? Can induce the nodulation. So it means it can go only this pathway. There is no bad effector to induce the plant defense and inhibit the nodulation. So in this picture, I just want to tell you that the specificity of the lysobium and the plant is also depend on the plant species, right? Some plant species, the wild type can make good symbiosis, but some plant species cannot make a symbiosis. But the reason, the mechanism why they cannot make good or bad symbiosis because of the type 3 secretion system. Okay. This is another example of type 4. This is the same thing, right? In the wild type, right, they can make nodule, have a good symbiosis, but when we delete the type 4 secretion system, this one make a bad symbiosis. It means that this lysobium requires the positive effector for type 4 secretion system. Okay, but for another system, in this lysobium, they have two copy of the type 4, but another copy that is no effect. Okay, right? And when we mutate both of them, because we mutate gene 1, copy 1, that's why they show the yellow plant like this. Okay, this is also the role of type 4 secretion system. And the application of the type 3 is not just only to make nodule. We found that, right? We found that when we inoculate the plant, right, with lysobium, and in the same time, we inoculate the fungus that produce the Cercospora leaf spot disease. You can see this is the disease, right? We inoculate the disease on the plant leaf, okay? In the non-inoculation of lysobium, you will see that a lot of plant disease symptom in here. But when we inoculate, right, the legume with lysobium like this, DOA9, this is the lysobium, and we inoculate the plant disease, you will see that the symptom of disease is reduced. Okay, it's reduced. It means that this lysobium can somehow activate the plant immune to protect the plant from the disease, from the fungus disease. And compare when we delete the or mutation on the secretion system, they cannot, the lysobium cannot send the effector to the plant. You can see that, you can see a lot of symptoms, right? So from this picture, it means that, from this research, means that some effector from the DOA9 that sent through the secretion system can activate the plant immune system and induce the plant to protect from the plant disease. Okay, so this is just another benefit of rhizobium, that we can use secretion system to protect the plant from fungus disease. Okay, so for the application for this symbiosis or type 3 secretion system, I think we can use the positive effector protein, right? Because when we know that some protein may produce, right, some protein may produce the positive interaction to induce the nodulation, right? So if we can produce this protein, right, and then we can supplement, we can supplement in the inoculation or co-inoculation with lysobium, it will increase the symbiosis efficiency. Okay, so this is the first application that we can make it in the future. Okay, and the second one, once we have the knowledge on tight restriction system or effector, how can we apply, right? We can apply as the co-inoculation to promote the symbiosis, but using the positive effector protein to produce. Another one is we can select some effector that can induce the plant immunity, right? And then we can just spray it on the plant, okay? And this effector can induce the plant immune. So that's why this one can induce the plant immune, for example, the induced systematic resistance system. So this one, if we spray it with this effector, if we have some fungus or disease come to attack, the plant already plumbing the immunity, right? It's already plumbed the immunity, so it can protect the disease. Okay, so this is the future application for the symbiosis. Okay, another application for symbiosis is the nitrogen fixation, right? Nitrogen fixation, lysobium, right? We know that it can produce the enzyme called nitrogenase. This enzyme very important. We found just only in bacteria. This enzyme can make the nitrogen gas, right, into its nitrogen gas and change nitrogen gas to be ammonia, right? And this ammonia, the plant can uptake as the fertilizer, okay, as the end fertilizer. So this enzyme is very important. We cannot see this enzyme in fungi. We cannot see this enzyme in plant. We cannot see this enzyme in plant or in other organisms, just only in bacteria and only in some bacteria too, okay? And lysobium is one of bacteria that can produce nitrogenase enzyme and fixed nitrogen. And the gene that can produce this enzyme we call NIF gene or fixed genes. Actually, this is not only one gene to produce this enzyme. You will see this enzyme is very complex, right? It's very complex. It contains so many components, right? That's why they require so many NIF genes, like NIF J, NIF H, NIF D, NIF K, right? So many NIF in here to encode the protein and produce a complex nitrogenase enzyme like this. Okay, so this enzyme catalyzes the reaction of changing nitrogen gas to be ammonia. Okay, so this is in our research, we try to find another NIF gene that maybe it's not the structural gene, it's not the gene that to produce the enzyme, but it's the gene that produce, we call homocitrate. Okay, so this gene we call NIF-V. In our lab, we call NIF-V. This is another type of gene that very, very important, and we found that this gene is very important for nitrogen fixation in the free living and in the symbiosis in lysobium. Okay, so in NIF-V, right, homocitrate will be as a cofactor for enzyme nitrogenase. It means that only a nitrogenase, they cannot function. They need the homocitrate as a cofactor. And this homocitrate produced from NIF-V. So it means that without NIF-V, they cannot produce homocitrate and the nitrogenase cannot function. Okay, so this is the importance of NIF-V. And in our research, we found that, okay, in some rhizobium, right, in some rhizobium, they can fix the nitrogen in the free living form. What does mean free living form? It can fix the nitrogen without symbiosis with plant, right? Normally, when we talk about the nitrogen fixation, lysobium will fix only in the nodule, in the plant nodule, right? Because in the plant nodule, it has the good condition, like low oxygen, to fix the nodule, to fix the nitrogen. Very small amount, a few lysobium can fix nitrogen outside the nodule. We call free living nitrogen fixation. And in our lab, we found 2 rhizobium can fix nitrogen outside the nodule, right? It's the lysobium SUT92 and DLA9. So you can see that in free living, right, they can fix the nitrogen, right? You can see the amount of acetylene reduction assay. This is just the activity of nitrogen fixation, right? But when we make the mutation of NIFV, NIFV does not function. You can see that the level of nitrogen fixation is reduced, right? It's reduced, significantly reduced. It means that this gene is very important. But once we supplement the homocitrate, it's just to prove the function, right? Once we complement, we add homocitrate, just the chemical compound, just add it into the mutant. You will see that the nitrogen fixation is increased, right? It's increased. So in this case, it means that homocitrate is very important for lysobium to fix nitrogen in free living form outside the nodule, right? They need the homocitrate, and homocitrate is produced from NIFV. Okay, so this is the free living form or fixed nitrogen outside the nodule. And how about inside the nodule? Inside the nodule, the NIFV is also important. Some plant cannot produce homocitrate, right? But some plant can produce homocitrate. It means that lysobium without NIFV can make the nodule and make the symbiosis, nitrogen fixation with the plant, but the plant will provide the homocitrate to lysobium. But in this case, this is the Achinomini americana, this is the name of the plant. This plant, it seems they cannot produce homocitrates for lysobium. That's why when we use the lysobium that make the mutation on NIFV, the plant cannot fix the nitrogen. So you will see that this is the very low growth of the plant. But similar to DLA9, right? When we make a mutation on NIFV, it reduces the nitrogen fixation. So from this slide, I just want to show you that not just only the NIF gene or fixed gene that encode to produce the nitrogenase enzyme. We need the NIFV that produce the homocitrate. And this homocitrate is the cofactor, and it is very necessary to make the nitrogenase enzyme function in the free living and in the symbiosis. Okay, so this is the mechanism. All right, so that what I talk is about the lysobium, okay, the symbiosis lysobium that can make nodule and make the nitrogen fixation for plant. But actually, when we talk about the bacteria or PGPR that can promote the symbiosis between plant and bacteria, there is another type of bacteria we call cyanobacteria. Okay, this cyanobacteria, this cyanobacteria can symbiosis with azolla. I'm not sure in Indonesia, are there any azolla like this?
Yes, we have.
Yes, yeah, okay. So this azolla contain inside the leaf, right? You will see the cyanobacteria like this. And this cyanobacteria, right, in the heterocyte cell, the biggest cell like this, is contain nitrogenase enzymes, right? The same nitrogenase enzyme as we found in the lysobium or in lysobium. And this one can fix the nitrogen to the azolla. So the nitrogen can accumulate in azolla. Okay, so this is another type of plant, not the legume. Okay, so this azolla in Thailand, we normally apply in the rice field. Okay, so we just grow it in the rice field, because in Thailand, when we grow the rice, we put some water. It's not the dry land. This is the wet land. So we put some water, and once this cyanobacteria fix the nitrogen, right, and accumulate in this water fluid or in azolla, we just mix it up, this plant into the soil. It's like a green manure, right? So the nitrogen from this azolla from this plant will be transferred into the soil. And then once later we plant the rice can uptake the nitrogen from this green manure when we mix it up in the soil. Okay, so this is just, it's not directly promote the plant, it's not directly promote the rice growth, but we just use the cyanobacteria that symbiosis with azolla to be used as a green manure, and use this green manure to be indirect promote the plant growth, promote the fertilizer to rise. Okay, so this is the another type of the symbiosis we call cyanobacteria and solar plant. Okay, so this is the PGPR in the symbiosis type. Okay, so are there any questions before we go to the second type of root associative? Are there any questions from students?
Not yet, not yet.
Not yet.
Maybe
Okay.
later after your presentation.
All right. Okay. Okay. Then continue to the second types of PGPR. We call root associative, right? This one, if you still remember, this is the bacteria that just stay nearby the root or just attach on the root surface, but it's not go inside the plant tissue. It's not make the nodule, right? Just associate or attach on the plant root. Okay. So this is the picture that show you. In our group, in our lab, we try to prove some PGPR, right? For example, Pseudomonas SUT19. This PGPR, we just tag with the GFP gene. Okay, so it means that this PGPR can show the green fluorescent protein like this, because you would like to prove that it is true that the PGPR will attach on the root surface, and how much is attached after we inoculate. Okay, and this one is the plant root. This is the foliage corn, right? The foliage corn. You will see that after we inoculation, these green are the bacteria. This is the PGPR that go and attach on the root surface. So this one confirm that the first property of PGPR, they should attach on the root surface, because once they produce any benefit, for example, nitrogen fixation or produce phytohormone, they can produce this nutrient nearby the root and the plant can uptake it, right? So it means that the first criteria, the bacteria should locate it nearby the root or attach on the root surface. And how the bacteria can attach on the root surface? We found that the bacteria can produce the biofilm, right? The biofilm, right? We have some some experiment to check the biofilm formation. You can see that some bacteria produce a lot of biofilm, some bacteria produce less biofilm. So the biofilm is like a polysaccharide that used for bacteria to attach on the plant surface like this. So this one we use the scanning electron microscope to see that, okay, the bacteria is attached on the root surface. Okay, so how the PGPR in the root associative type promote plant growth? Okay, the first function or first mechanism is the phosphorus or potassium solubilization. Okay, phosphorus or potassium solubilization. Why we need this function? Why we need this function? This function is like the bacteria, right? The bacteria can just produce some acid or produce some enzyme to solubilize, right? Solubilize the phosphate or potassium that normally is in the insoluble form, right? Normally this P or K will be attached with some metal ion or some mineral and make it not dissolve, make it precipitate in the soil. It cannot dissolve into the water. I'm not sure if you know that the plant can uptake the nutrient only when the nutrient dissolve in the water, right? The plant can uptake only when the nutrient dissolve or solubilize in the water, right? Because in the soil has the water, so the plant, the nutrient must be solubilized or soluble in the water, and then plant can uptake. But in this case, if the potassium or phosphorus bind with some mineral, this one will precipitate and cannot dissolve in the water, and it can precipitate in the soil. So in this case, the plant or the root cannot uptake the phosphorus or potassium. Okay, so this is what we need some bacteria to produce the enzyme or to produce some organic acid and dissolve this precipitate phosphorus or precipitated potassium to be dissolved in the water. So if you see this picture, we grow the bacteria on the medium that containing phosphate that in the insolubilized form or precipitate form. Okay, so you will see the obidity like this, right? The dark like this. But once we put the bacteria that can produce some acid or some enzyme that can dissolve this precipitate phosphorus, you will see the clear zone, right? You see the clear zone. It means that in this clear zone, the phosphate change from the precipitate form to be the solubilized form or dissolved in the water. So once this bacteria come to this mineral or precipitate, phosphorus or potassium, they will dissolve and then produce the phosphorus or potassium in the solubilized form, soluble form. And then the plant can uptake it and then use it for plant growth. And as I asked you in the beginning, why we need this one, why the phosphorus or potassium stay in the precipitate form. If you see this picture, okay, this picture show you the nutrient solubilization in the soil at different pH. So this is the pH of the soil, right? In this part is the acidity, acid soil, right? And in this side is the alkaline soil. You will see that in each nutrient, they have different ability to dissolve in the soil, different soil pH. For example, most of the like nitrogen, phosphorus, or potassium, right? Most of the solubilization activity or soluble activity is less when stay in the acidic soil or in low pH soil, right? You will see that less nutrient available, right? Same thing in the alkaline soil, right? Some nutrient is less, like iron or manganese. In the neutral pH soil, right? Neutral pH soil like pH 6.5 or 7, most of nutrients are available or soluble form. That's why when we tell the farmer, okay, try to adjust your soil to be neutral soil because the plant can uptake most of the nutrients because the nutrients are in the soluble form. But if we grow the plant in the acidic soil, even we put the fertilizer, chemical fertilizer, phosphorus or potassium in the soil, in the acidic soil. In this soil, the potassium or phosphorus will not dissolve, right? It will be in the precipitate form like this. That's why if we grow the plant in acidic soil, if we use the bacteria or PGPR that have the ability to do the phosphorus or potassium solubilization, it can help to produce the soluble form of P or K and make the plant to uptake and then promote the plant growth. Okay, so this is the first function of the PGPR in the root associative, right? So once they stay like this, they can dissolve the P or K and then make it available for plants. All right. And the second function is the biocontrol. Okay, the biocontrol. Some PGPR in this associative can produce some activity. It can produce sometimes like antibiotics or compound or some enzymes that can degrade or inhibit, right? Inhibit the growth of pathogen, right? For example, if we grow the fungi in the middle of the plate like this, normally the fungi will continue grow, right? Continue growing. But if we put the bacteria, right? The bacteria in like this and on the side like this, if the bacteria can produce some activity like enzymes or produce some antibiotics that can inhibit the fungi growth, you will see that the fungi cannot grow across the bacteria. Or it can produce the clear zone like this, because during this clear zone, there is some antibiotics activity or some enzyme that the bacteria produce and inhibit the growth of fungi. So that's why this type of PGPR can also inhibit some plant disease too. Okay, this is a biocontrol. And in our lab, we found some bacteria that can inhibit the the fungi called Aspergillus niger. This is the very big problem in Thailand when we grow peanut, right? This fungi can produce aflatoxin. If you know aflatoxin, this is the Aspergillus niger. And this Aspergillus niger also cause the root rot disease in peanut when we grow peanut in the field. So we try to reduce the symptom, but most of the time the farmer use the chemical compound like a fungicide, for example, the carbendazim. This is somehow toxic if it still remain on the root or on the pot, right? We have to eat it. This one can be toxic to human or to people. So we try to find the PGPR that can inhibit the Aspergillus niger and then use instead of the fungicide. And in our lab, we found two PGPR, A62 and A20. This is the bacillus can inhibit the growth of Aspergillus niger. And when we apply together with the Lysobium, you can see that it can show the good modulation and in the same time produce some activity to inhibit the Aspergillus niger too, right? So it's quite good when compared with the inoculation with Aspergillus niger alone or use the fungicide. So this is another function of PGPR. Okay. The next function is the function of the bacteria that can produce plant hormone. Actually, it's not just only IAA. They can produce the IAA, cytokinin, gibberellin, right? They can produce, but it does not mean one bacteria can produce all type of hormone, no. But one bacteria can produce one type of hormone, or at least two, okay? So this hormone is very important for plant growth. For example, IAA is very important for the root, right? Root growth or root elongation. So some bacteria can also produce this one. Okay, this is the example that when we apply the PGPR, right? PGPR, in this one we call it PGPR SUT1. When we apply this together with compost, it seems that it produce a better benefit to plant growth, right? Compared to use compost alone or use the PGPR alone. So this one is tell us that actually the plant growth, when using just only PGPR is not enough. It's better to apply PGPR together with the compost or organic fertilizer. Okay, so this is the example. This is another example. We try it with the lettuce, and we try to see that whether this is the effect of organic fertilizer or not that can promote the plant growth. And it's yes, when we apply the compost just only like this, but when we apply the PGPR with the compost with just only 100 grams, this is also 100 grams, it seems that the PGPR has some effect. When we add the compost or organic fertilizer, just 100 grams, we can see a little bit better. But when we add more compost, right, 200, 300, or 400 grams, it seems that the effect of plant growth is come from the compost more than PGPR. But actually, they may work together to promote plants. So from this slide, I would like to suggest that when we use PGPR, it's better to use it together with the organic fertilizer. Okay, and another function we found that when we use the PGPR, right, PGPR associated, this one is Bacillus valerensis S141. This one, when we co-inoculation with Lysobium, right, this one, Lysobium SUTN92. This one, somehow they can make a bigger nodule and make a better symbiosis. We don't know yet how this valerensis or this bacillus make the mechanism to promote the SUTN92 to produce a bigger nodule, how or why, we don't know, but we just found that they make a bigger nodule and promote a better nitrogen fixation. Okay, so this PGPR, another application we can co-inoculate with Rhizobium. Okay, and I would like to show you another function of PGPR. Most of the students or most of the people think that PGPR can just produce a compound that dissolve in the water or dissolve in the medium. But actually, some PGPR can produce the volatile compound. And this volatile compound in the gas form, not gas. So the volatile compound can support the plant growth. You can see from this experiment. Okay, so in this experiment, we put the bacteria PGPR in the medium that separate, right? This is a plastic. This plastic separate the medium of the bacteria grow and the medium that the plant grow from each other. It means that if the bacteria produce or secrete some compound into the agar, this compound cannot pass this plastic into the plant. But how the plant can promote when we put the bacteria? It means that this bacteria can produce some gas or volatile compound, and this gas can also activate or promote the plant growth, right? Compared to this one without PGPR, the plant does not grow well. There is not many roots, right? So I just want to tell you that not just only soluble compound, PGPR somehow can produce the gas phase or volatile compound to support plant growth too. Okay. Another property of PGPR, so you see that PGPR has so many activity or function more than lysobium. Lysobium we focus on nitrogen fixation, right? But for PGPR in the associative, they produce a lot of function. This is another function. Some PGPR in root associative type can produce the siderophore. Siderophore is the compound like this. There are so many types. This is the type of compound. For example, the hydroxymate, catecholate, or carboxylate. This compound of siderophore can bind with the iron. Yes, you see that this compound will bind with the iron. So why this is benefit? Because most of the time in the soil, there is like we have the problem of iron deficiency, right? That we do not have enough iron in the soil, or the iron bind with some other mineral in the soil and precipitate and cannot use by bacteria or plant. Okay, so if we apply the PGPR that can produce this compound or siderophore, it can bind with iron, right? It can bind with iron and make it available for plant to uptake, right? It means that the plant, when we inoculate with the PGPR that produce siderophore, the siderophore can bind with the iron in the soils, and the plant has more chance or has more availability to uptake the iron into the plant, right? It means that more nutrient can provide to the plant. Okay, so this is the siderophore with the iron uptake. Another function of PGPR we call the function of ACC deaminase enzyme. This function is very important right now because we found that with the changing of climate, right? Climate change. Sometimes it's very hot, sometimes flooding, sometimes cold, sometimes I don't know. It's the abiotic stress, right? From the environment stress. This stress can affect a lot on the plant growth. Okay, and we found that some PGPR can reduce this stress. How can it reduce the stress? I have to tell you that normally when the stress response in the plant, it normally comes from the ethylene. The plant will produce hormone called ethylene signaling. So this hormone normally is produced in the very low level, just to produce to activate the cell growth, the cell senescence. But once the plant responds to the stress condition, the environmental stress condition, somehow this pathway of the production ethylene is activated. The ethylene is produced a lot, and very, very high amount of ethylene will be produced during stress. And this stress response will induce the plant to like fall down the leaf, to stop the growth, to stop the root elongation for a while, because this is the stress condition. They try to stop the growth. Okay, so if the ethylene is produced a lot, it will induce the stress response and stop the plant growth. And this will affect the yield or the whatever plant that they will produce after. Okay, so how to reduce the plant growth? to reduce the plant growth, one way is to reduce the ethylene production, right? To reduce the ethylene production, because the ethylene is the plant signaling to induce the stress response. So how to reduce the ethylene? We have to know that the ethylene is produced from the precursor called ACC. ACC is the precursor that used to produce ethylene using the enzyme called ACC oxidase. This is occur in the plant cell. But we found that some bacteria, some bacteria can uptake ACC from the plant and produce the enzyme called ACC deaminase. And this enzyme can change the ACC to be ammonia and alpha ketobutyrate. This is the nitrogen source for bacteria growth. It means that the bacteria that contain this enzyme can use ACC as the nitrogen source and growing. But the good thing is, if we inoculate this bacteria with the plant, if the plants get the stress condition from outside, the ACC will be reduced, right? Because the bacteria will uptake ACC. And once the ACC reduced, the ethylene signaling will be reduced too. And once the ethylene reduced, the stress response will be reduced. So that's why when we inoculate the bacteria PGPR that containing ACC deaminase activity, and if they stay with plant, they can reduce the plant stress. This is the experiment that we prove it. Okay, we try to find out the bacteria that contain the ACC deaminase, right? For example, ACC 1, 2, 3, right? We found 3 strains. And we test it in the water stress condition. You will see that we co-inoculated it with the rhizobium, okay? You will see that in the stress condition with only rhizobium alone, when it encounters the plant stress, for example, the water stress of water deficit, the deficit condition, the plant cannot grow well. But with the ACC deaminase containing PGPR, right, co-inoculation, the plant can continue growing. But I have to tell you that the stress condition must be in the short period. For example, 1 week. But if it's longer than like 1 month, the ACC deaminase cannot help you. Nothing can help the plant die. The ACC deaminase activity can help to reduce the stress once the stress occurs in the short period, like 1 or 2 days, or at least in 1 week. Okay, this is to make the stress, to make the ethylene keep it in the low, and the plant can still continue growing. But in this case, the plant, when encounter the stress condition, they stop the growth. But this one, they can still continue the plant growth. Right? 1 week is also effect. Okay, and in our lab, we know that, okay, the PGPR that contain the ACC deaminase activity or this enzyme is very important to be used to reduce the plant stress when we grow in the field, right? So how to improve this activity? Because normally when we try to screen the bacteria from the soil, when we try to collect the sample from the soil, from the field, normally we found the activity of the ACC deaminase or PGPR only 1 to 2 micromole, right? Micromole, very low. We would like to know that if we improve it to make the ACC deaminase activity more than 3, what happened? Okay, what happened? So we try to do it by using the technique called adaptation. Adaptation means adaptation. We try to put the bacteria or grow the bacteria in the medium containing ACC. This is the substrate. But in the ACC, we start from the lower concentration, right? We start from the lower concentration. So when we put the bacteria and start culture, the bacteria, if they can use the ACC, they can grow, right? They can grow, they can start growing like this, make the turbidity, okay? And then after that, we take the culture from this flask, increase or inoculate into another flask, another medium, but we increase the concentration of ACC. Okay, when we do like this, continue to do like this and increase the concentration of ACC, right, to be more than 3 millimolar of ACC. Okay, so in this case, we hope that the ACC deaminase of this bacteria will be increased naturally by adaptation. Okay, by adaptation. Okay, but we don't know how they adapt. But we know that okay, they must increase the activity from one level into the another level. Okay, so this one is the one strategy we call adaptation. And another strategy that we want to use it to improve the ACC deaminase, we want to make GMO, right? Because we found that when we use adaptation, the activity increased from wild type, not so much, not so many, right? Just maybe only one fold, not so many. So we want to know if we increase the activity of ACC deaminase more than 10 times or 20 times, what happened. So we use the technique called genetic engineering. So you may know already, we just clone the gene, right? ACD gene, the gene that used for making enzyme ACC deaminase, and clone it into the plasmid and transfer into the lysobium. Okay, so this one become the GMO. Okay, so this one I compare, I show you the activity. So this is the wild type. Activity is already 3.8. But when we make the adaptation, right, as I show you in this picture, the adaptation technique can increase the activity of ACC deaminase from 3 to 5. So just a little bit, right? But when we try to use the GMO technique, so we clone this gene into this vector and then transfer to this bacteria to be GMO. This GMO bacteria produce the ACC deaminase activity about 10 times, right? More than the wild type, this is 34, so about 10 times. So we would like to know that if we increase like 10 times more than usual, what happened? Okay, so we check again by inoculate these bacteria, right? Compare between wild type, the adaptive, and the GMO one, okay, into the mung bean under the water stress or water deficit condition. And you can see that this is normal condition. In the normal condition, there is no difference between the nitrogen fixation or the ethylene production, right? But in the drought condition, you will see that the nitrogen fixation is very high when we use the GMO. That means that the bacteria can reduce the plant stress, right? But when we use the wild type or the adaptive strain, yes, it can increase the nitrogen fixation compared to non-inoculation, right? But the level is still low compared to the GMO, okay? And when we check the ethylene production, right, we know that if we use the bacteria that produce a lot of ACC deaminase activity, the ethylene in the plant should be reduced. And yes, we found in the non-inoculation, in the drought condition, ethylene produced very high, very stressed. But when we use ACC deaminase wild type, right, or adaptive, it reduced the ethylene production. And if we use the GMO, it significantly reduces the ethylene production, and that's why it can improve the nitrogenase or the nitrogen fixation better than the wild type or adaptive strain. But however, in Thailand, right, we cannot use GMO in the field yet. So in this case, we cannot make like 10-fold, 20-fold higher activity, right? So we have to still keep using the adaptation technique to continue, right, increase the adaptation of the ACC deaminase activity to make it more and more, okay? Because the GMO is not allowed to be used in Thailand. So adaptive techniques still be a good choice for us to further improve the strain, okay, to be the inoculant. All right, so this is the function. So you will see that for the root associative PGPR, they produce so many functions that can promote plant growth or support plant growth under the stress condition. The last one is the endophytic bacteria. Okay, so this one is the bacteria that stay inside, as I told you. It's go inside the plant tissue, right? It can stay in the intercellular space of the cells, or inside the cell, in the vascular, or in the leaf, in the stem, in the root. Okay, wherever they can stay. So this bacteria is endophytic bacteria, and if it is PGPR, it means that they should promote the plant growth, right? So we found that we found the S2TN92. This is the lysobium that I show you from the beginning. This lysobium can be lysobium that can make nodule with ligum. And we found that this lysobium can be endophyte in the rice plant, in the rice. And we found that this lysobium in the rice, it can fix the nitrogen. Actually, we have another lysobium that can be endophyte and fix a lot of nitrogen. Oh, sorry, this is the IAA production, a lot of IAA production. They can produce ACC deaminase, right? They can produce ACC deaminase, and they can fix the nitrogen. This is occur in the rice, in the free-living form, not in the nodule, right? So this is, I'm going to tell you that lysobium, some strain can be endophyte PGPR. So they can stay inside and promote the plant. For example, this is in rice, they can promote by produce IAA hormone. They can promote by produce ACC deaminase activity, or they can promote by fix the nitrogen. Okay. And we found something more interesting. So this is the picture of the S2TN92 when they stay inside the cell of rice. Okay, so they stay inside like this. We found that if you notice, the cells of bacteria is very long. It's very long compared to the normal cell when we grow in the liquid culture. When we grow in the liquid culture, it's quite short. And this size of bacteria is longer when it stays longer in the plant cell. I don't know why. And we found that this elongation of the plant cell is correlated with the nitrogen fixation in the plant cell. So this one, we check the expression of NIF gene. This one is NIF-H. If you still remember, NIF gene, this gene is used for making enzyme nitrogenase, right? So if this gene in lysobium expressed, it means that it produces nitrogen. It's produced nitrogenase to fix the nitrogen. And we found that this gene is expressed in the root, okay, at 1 month. But later on, we found very low expression of this bacteria. But we found this expression in the leaf sheet. And even at the later day, like 28 days, 70 days, the highest is at 70 days. The nitrogen fixation or the NIF gene is expressed very high. It means that this lysobium may move from the root, right? From the root at the beginning. Because when we inoculation to the root. The bacteria may move or locate from the root and then move from root into the leaf. And once they stay in the leaf, they can produce the NIF or nitrogenase activity to fix the nitrogen. So this one proves that this lysobium can stay in the leaf and have the nitrogenase activity even at 70 days after inoculation. Okay, and this elongation, yes, if you can see, this is the in the culture medium, liquid medium, the cell size is very short. But once we found it in the rice and then we extract it, you will see that the size is quite long. It's quite long like this. And it's very interesting when we found that the size of the cells, you can see the bar chart is the size, and the dot is the nitrogen fixation activity, nitrogenase activity. So you will see that at this is I use the DI water, and this one use the light extract, right? This one when use the light, extract to induce the elongation of the cell. The cell size is increased from 7 days to 28 days. The cell size is significantly increased, elongate. And the nitrogen fixation activity is also increased. So it means that the enlargement of the cell size is correlated with the nitrogen fixation activity. So this is very interesting for us to study why or how this lysobium can stay inside rice, can fix nitrogen, and the fixation of nitrogen is correlate with the cell elongation, which we never found before. Okay, so this is the endophyte. And the next slide, I would like to tell you about how we apply this PGPR, right? This PGPR for commercial or for application. As we know that the SUTN2 lysobium, this lysobium is very interesting. We found that this lysobium not just only to be the endophyte in rice. We found that this lysobium can be endophyte in vegetable, in eggplant, in chili, in tomato too. This is very interesting. So we try to apply this by using this lysobium together with the PGPR. If you still remember S141, this one when we put together, somehow they can promote the bigger nodule of lysobium, of nodule of the plant, right? So it seems that S141 can promote SUTN2, and S141 somehow can also promote the plant growth itself too. Okay, so we apply to make a product we call Smart Soil by use the biofertilizer of S141 together with SUTN2 and use trichoderma. Okay, I think everyone knows trichoderma. Trichoderma is the fungus that can protect the plant root from the root rot disease, right? So this one we just mix it together and then put it in the planting material, and we call it Smart Soil. So it means that this Smart Soil not just only provide the nutrient from the soil, but we still have some bacteria to promote plant growth, right? And we can apply use with not just only legume, we can use with chili, we can use with tomato or another vegetable as the endophytic PGPR. Okay, and also we apply the trichoderma as the biocontrol. So it means that the Smart Soil can be both PGPR to promote plant growth, can be the biocontrol, and also the nutrient from soil can support the plant growth. Okay, so this one is very popular, and this one, the SUT farm, the university, our university take this knowledge and then sell this product to the farmer. Okay, this is the picture that I show you, that we compare the Smart Soil with the normal soil that they sell in the market. You will see that when using the Smart Soil, it's produce a better growth for the plant, right? This is the bean, right? This is the yard long bean. This is the eggplant, right? This is a chili, for example. Okay, and in Thailand, as I told you, the production or the selling of the PGPR is not so popular because of the very strict regulation from the Thai government, very strict regulation, very strict laws. So as you know that when we use the PGPR as biofertilizer, this is the living organism, right? Living organism. They will produce so many functions, as I told you. The function of hormone production, the function of nitrogen fixation, produce siderophore or biocontrol. This function will be working only when the cell or bacteria is still alive. Right? Still alive. It means that when we sell the product, right? We sell the product. This bacteria, this PGPR must be alive in the package. And this is the big problem for our industry. If we want to produce this product in the commercial scale, the problem is we cannot keep this product in a long time. Sometimes, for example, this smart soil, we check the shelf life of the bacteria in the soil. Only 3 months, then the number of cells reduce. So we recommend that if you buy this one, if you produce this one, we have to finish it or use it before 3 months. Then otherwise, the number of cells will be reduced because some of them die. And this is the big problem. And as I told you, the Thai regulation, the Thai government, the law is very strict. They say that for PGPR, you have to have at least 10 to power 6 cells per gram. Right? For example, if you take 1 gram of soil or 1 gram of product, you have to have at least 1 million cells and 1 million of living cells in the product. And most of the time when they sell it, they keep it in the outside the building. They're facing with the sunlight. They're facing with the high temperature. This high temperature can also heat up the product, right? And then somehow can kill the PGPR. So the number of cells normally reduce, right? Normally reduce. And once the police come and check the product, they say that, oh, this is wrong, right? Because the number of cells is less than the regulation, is less than the standard. Okay, so this is the big problem when people who want to do the business in PGPR production or biofertilizer production, they try to solve the problem. For us to making the smart soil, we try to solve the problem by put more cells at the initial. The standard they make 10 to power 6, right? But for us, we try to put it 10 to power 8 or to 10 to power 9 cells per gram at the beginning. So that's why we still have a longer time to, like, when the time passed, the cell number is reduced and the number is still over the standard 10 to power 6. And once the police come and check, oh, okay, it's still over the standard, it's fine, right? And the another problem of the Thai regulation when we selling the biofertilizer product or PGPR product, they define the regulation. They define the definition of PGPR as a single inoculum. For example, if we want to sell bacillus, it must be only bacillus in the package. If we want to sell Pseudomonas, it must be Pseudomonas in the package, only one. But nowadays, but nowadays you may know that we cannot use just only one PGPR for one product, for one plant to support growth. Normally, we have to co-inoculation more than one single type of bacteria, right? But in the Thai regulation, they tell us that only one type of bacteria for one package, for one product. So this is the very old regulation that I think we need to change it, right? And as I told you, the GMO is not allowed to be used here in Thailand. And another one thing is, you may know that the PGPR, actually they can produce many compounds that very useful for plant growth, right? For example, as I told you in the beginning, some rhizobium can produce the effector protein to promote the plant immune and promote the resistance of the disease, right? So if we can produce the effector protein as a product, like a compound, and selling for spraying the plant, this can be another line of product. But in Thailand, there is no regulation, there is no laws to accept this type of product, right? We cannot sell the compound like this. We cannot extract the compound and sell it as a product. You have to sell the microbial, the bacteria like this. So this is the problem, okay? So that's why, because of the strict regulation, the old laws of Thai government, in Thailand, we do not have many company selling the bioproduct or PGPR. This is the biggest one that I show you, the SV group. Okay, this one is the biggest group of the company that selling the biofertilizer product. They sell the PGPR, they sell the dichroderma, they sell even the mycorrhiza. This is another type of biofertilizer, but this is the fungi. That's why I did not tell you about this today. Okay, but as I show you, they just produce one type of PGPR in one product, right? One type of bacteria, one type of fungi in the product. So they just only one company in Thailand that this is the biggest one to help to sell the product and help the farmer who working with the organic farm. Okay, so this is the problem in Thailand. And before I finish the class, I would like to tell you that actually nowadays for the industry of biofertilizer production of the company, nowadays it's not just only I would say small business. This is a big business now. If you see this company AgBiome, this is the international company AgBiome. You can search in the internet. This AgBiome, this company try to collect most PGPR. They collect a lot of PGPR from many areas in the world. You will see that they have a lot of collection, and actually they have a trillions of microbes. You will see that this type of company they're gonna have more influence for us in the future, because they try to collect the most efficient PGPR from the nature, from the natural source. And once they collect it, they get it to prove the function. They make from a trillion microbe, they might get 1,000 strains, right? They can produce, they can test it in the greenhouse to make sure that this PGPR or this bacteria is good for plant promotion. And from this, they have the activity to test it in the greenhouse, in the field, and finally produce the biological product. This is the normal line that, okay, in Thailand we do this too, right? In Thailand we do this too. And I am sure in Indonesia also doing this line. But AgBiome do more than that. They sequence this PGPR, the best PGPR they collect, the good PGPR they collect, they sequence their genome. So once they sequence their genome, they know the DNA sequence, they know the candidate gene, that what gene can make a good IAA, what gene can make a good siderophore, what gene can make the good nitrogen fixation, right? So they have a big data of gene, right? The trait, the genomic data. And this gene will be very important for them to make a GMO later on. Because in USA, right, or in another country, they're allowed to use the GMO. So once they know the gene that produce the efficient function of hormone production, of the siderophore production, right, they can clone this gene, right? Or they can improve the activity of the enzyme. So in this case, you will get the super bacteria, super GMO biofertilizer. Okay, so this is the future trend. So this company is working a lot on this one. You will see that they try to collect a lot of microbial strain in their collection. Okay, and based on their sequence, not just only the hormone production gene or nitrogen fixation gene, they know even the gene that used for produce some enzymes or some antibiotic that can inhibit the fungi or inhibit the plant pathogen. So in this case, they can produce the enzyme or they can produce the antibiotics as a compound and produce it as like a product to spray. So like this one, they can produce to sell it as a compound to control the insect. Okay. Yes, there are so many pesticides that synthesis from the gene, from the good bacteria. So this is considered as the biological compound because it's produced from bacteria, right? It's not the chemical compound. So they plan to sell this one, right, in the market. So you will see that in Thailand or in our Asian country, we still working on the old regulation. We're still working on just only the cell bacteria. But in the international company, they're working on the gene. They're working on the how to say the trait that more than just nutrient. It can be the biocontrol, or it can be the insect control or fungus disease control. Okay, so this is the product that they are going to make, right? They sell the chitinase enzyme, they sell the gelatinous enzyme, for example, or they sell some like a compound that can be used to kill the bacteria or to kill the fungi, right? So this is the new product. Okay, new product. Okay, so for conclusion, okay, so it takes a lot of time, so sorry. So in conclusion, today I told you about the definition. So try to think about this. I told you about the definition and types of PGPR, right? So we have symbiosis, right? Mainly I talk about the rhizobium. We have the mechanism of modulation, why it's specific, right? Why it can fix nitrogen. We talk about the type of root associative bacteria, right? This one have a lot of function, right? They can just associate on the root, okay, and have a lot of function to promote plants. And the last type is the endophytic bacteria. This one can stay inside the plant tissue and can promote the plant growth. Okay, and we're talking about the mechanism and the function of PGPR that can use for promote the plant growth promotion, and how to apply the PGPR for agriculture and in the industrial level, right? As I told you that now the big company, they just looking for more bacteria, okay? Not just only one single cell. They look about the microbiome engineering, try to produce a mixture of culture inoculum, not just only single inoculum, okay? And also produce some biostimulant compound, okay? And yes, law and regulation are needed to be improved in Thailand. Okay, so thank you very much for your kind attention.
Okay, thank you very much, Janop. That's very interesting and insightful presentation from you. And I think you really can sum up one semester lecture in your one presentation.
I hope
because this is because you really give so broad and deep explanation on what is plant microbe interaction. Okay, so everyone, if you have any question, you can raise your hand, or you can write in the chat room. And in the chat room, there are already many questions.
Wow, okay, I can see now. Yes, thank you for the questions, everyone. Yes, okay.
So,
okay, if you have questions, okay. My name is Inham Aris Wandi. Okay. From the bioenergy study program, I want to ask if there is a condition where a PGPR successfully enter the plant root but fail to form nodule. If there are, what are the cause and factor that influence the success of formation of nodules? Yes, very good question actually. Okay, I will show you my own experience. I use the rhizobium, right, to make the nodule on soybean. Oh no, sorry, on mung bean. I try to use it in many locations in Nakhon Ratchasima, in my province where our university is located. And we found that some location is work. The rhizobium make good nodule. Some location it does not work. No any nodule or very, very less amount of nodule. And we found that the first factor is the soils. Some soils contain very high nitrogen content. It already has nitrogen content in the soil. So once the soil has nitrogen already, even we apply lysobium, the plant select the nitrogen from soil, because it does not take a lot of energy to just uptake the nitrogen from soil to plant, but it takes more energy to make nodule to give the carbon source to lysobium, right? So if the soil has high nitrogen content, it will not produce a nodule. This is the first one. The second factor is the environmental factor. For example, if you, I found one experiment, the student just sow the seed and inoculant in the noon, like daytime, very hot, very hot day, because they try to finish it and then go back to university early. And they say that, oh, I just sow the seed with inoculum in the noon or in the afternoon. And you know that in the afternoon in Thailand, how hot is it? Very hot, right? We found that we get very less nodule because most of the lysobium die during, in the soil. It takes about 1 or 2 days to get this mechanism until the lysobium get inside the nodule. And during this 1 or 2 days, if they face with the bad environmental factor like high temperature, the lysobium die. So when lysobium dies, they produce less number of nodules. Yes, and also the pH of the soil. So mostly it's about the environmental factors that affect the lysobium modulation. Yes. Okay. If you have more questions, you
Yes.
can. Yes. I am interested. This is from the Lomalio. Okay, Lomalio asks, I am interested about ACC deaminase activity of bradylysobium and its developed strain. Okay. What kind of genetic modification technique that you use to improve the strain that with high ACC deaminase activity? Okay, thank you. For the genetic engineering, actually in this paper we use 2 techniques, right? The adaptation, this one is we just continue growing bacteria in the medium and then try to a little bit increase the concentration of ACC a little bit more and more, right? This is not non-GMO. This is naturally changed. But for the GMO, we use the genetic engineering by using the plasmid, right? We use the plasmid, cloning plasmid, PMG103. And then we clone the gene. We call the gene ACDRS. This gene is used for produce the ACC deaminase. So these 2 genes were cloned, right? And then ligated into this vector. So you get the recombinant plasmid, right? So this recombinant plasmid has the gene for making ACC deaminase. And this gene will transfer back to bradylysobium. So this bradylysobium have their own one copy of gene on the chromosome, right? And they have more copy, about 7 to 8 copy, because this plasmid can make about 7 to 8 copies of the plasmid. So you have another 7 or 8 copies of this gene in the cell. So that's why you have 7 to 10 copies of ACC deaminase gene to produce ACC deaminase. That's why you have higher activity of ACC deaminase in the GMO strain, about 10-fold. Yes. So this is a technique that I use, but we cannot use this one in the field because this is GMO. Yeah. Okay. And for the question for Fernia, very interesting. I am Fernia, and your PGPR, SUT plus compost treatment show the same response with our PGPR consortium compost treatment that can increase the plant growth. If we can to know which is the main isolate that involved in this mechanism, may you suggest what method can we do? Okay. If I guess she would like to know that, how to know that which if we use many strain, right? If we use many strain
Yes.
or a mixed strain, or multiple strain of PGPR, and if you want to know which strain is the best or have the, how to say, the highest activity when among each other. Okay, this is very good question. We also try to do this in our research too. One thing is we have to consider on the number of cell that still located on the root, because one hypothesis is if the bacteria can promote plant very well, right, very well, the plant must produce something. This must be the positive interaction, even it's not symbiosis, direct in symbiosis, but the plant must all try to keep this bacteria nearby their plant, right? Because this bacteria is very good, they try to keep it nearby. So the number of cells nearby the plant on the root surface, the number of bacterial cells that show highest number, this is, we can hypothesize that this bacteria is the good PGR that can promote the plant. And the second thing is we have to prove the activity. This one we may have to use the genetic, use the molecular technique. For example, if we can tag the each strain. For example, in my case, I tag the PGPR S141 with the DS red. This is can produce the red color of fluorescent, and use the Bradyrhizobium use the GFP that produce the green color. Okay, so we tag this strain, but this one we have to do in the lab, and then we try to see that how this bacteria interact with the plant, how they locate on the plant. They go inside, they go to the plant leaf, they go to the stem or not. This is to make sure that once the plant grow, we can see the plant growth is come from our strain. Because if we did not see this strain, it means that they grow because of the compost, not because of our PGPR. So we have to make sure that these two strains still presents in our plant. Okay, so this is the second one, the persistence of the strain in the plant. The third one, we have to detect the expression of some gene. Right? We can decide, for example, if we want to, okay, if the rhizobium still can express the nitrogen fixation or not, we can decide the gene or the primer that can tag or can follow up the expression of the gene in this bacteria, and checking, okay, if this gene still express all the time, express low or high compared to another bacteria. So this is, I think this is can be a method that we can check the efficiency of this bacteria when we after we apply to the plant. Yes. Okay. I hope I answer your question. Okay. Another question from Kiera. Okay. Kiera asks, every plant, especially organic plant, have a root associative PGPR. Because from your explanation about symbiosis, I think it's only for leguminous plant. If it is also in other plant, what do we need to pay attention to if we want to do isolation, characterization, and screen a potential PGPR from organic plants? Is it from the edge of the plant or the sampling zone or what else? Thank you. Okay. Many questions, but this is very good. It means that you understand this one. Okay. First question. Every plant already contain the associative PGPR? I said yes, but it depends on the environment that they stay in the soil, right? And even in the good soil condition, we found the bacteria associated PGPR that can promote plant growth that have the PGPR property or function only 2 to 5% of the population. Only 2 to 5%, no? Very, very low amount, yes, to stay and have this function. So if we would like to isolate right? If we would like to isolate the good bacteria, good PGPR, we have to make the criteria that you want the lysobium or you want the associative PGPR or you want the endophytic PGPR, because the way of isolation is different. If you want the lysobium, you have to isolate from nodule because it makes sure that this one can make the nodule, right? You have to isolate from nodule. But if you want to isolate the associative PGPR, it means that you have to isolate the bacteria that attach very firmly, very tightly on the loose surface, right? So it means that you have to, remove some soil particle and take just only the bacteria that attach firmly on the loose surface. Because one criteria, as I told you, the PGPR must be attached on the root. So that's why if the bacteria have so many functions, the plant can get the nutrient or get the benefit easily, right? So if you want to get the associative PGPR, you have to remove the soil and get the bacteria that stay or attached on the loose surface. And if you want to get the endophytic bacteria, this one is stay inside. You have to do the surface sterilization on the loose surface. Make sure that there is no any bacteria on the loose surface. And after that, you have to glide the root, right? Glide the root and take the bacteria inside the cell, inside the tissue to grow, to isolate on the medium. And then after that, you will get several bacteria. Then you have to test these bacteria with the different ability. For example, can they fix the nitrogen in the free living form? Can they make the nodule? Can they make the hormone? Can they make the ACC deaminase or not? Then you have to test the PGPR trait before select the best one by testing with plant in the greenhouse in the field. Yes. Okay, so try to select from the lysosphere zone for the associative. Okay. All right. From Nafila, many studies show that inoculating plant with PGPR or treating plant with microbe to plant signaling compound may become effective strategy to stimulate crop growth. But in the level of application, it's very difficult to improve lysosphere colonization by PGPR inoculant. And then from mass production, based on your experience, how to establish the roadmap for commercialization of PGPR-based technology product. Okay. And what role of PGPR play to underlay food security in 21st century? Wow, this is quite politic. It's quite politic. Okay. Let's start with the first one. Yes, to apply in the level of application. Even me, when I apply PGPR in the field, not many times it works. Many times we fail to apply the bacteria. It does not show any plant growth promotion, and most of the reason is come from the environmental factors that affect the cell growth or the bacterial viability. So to do this, to make a commercial, the research on inoculant formulation is very important, right? The formulation is mean how to protect the cell from these environmental factors. For example, can we use the encapsulation technique, right? The encapsulation? Encapsulation is when you can use the polymer to encap or encode the bacteria, cover the bacteria. So in this case, the encapsulation or the polymer might protect the cells from the environmental factors that affect the viability of cells. Or some formulation they can add some additives compound, because some plant produce some toxic to bacteria. So for example, I have seen some lysobium soybean. Some soybean produce the seed exudate that's toxic to lysobium. and we can add some additive compound to bind or to detoxify this toxic. So I think the research on the inoculant formulation is very important to make the PGPR application to success in the future. Okay, and for the colonization, yes, right now the one single PGPR is not enough, right? To make it better in the natural soils, normally it's contain more than one bacteria, right? It's more than one type of bacteria. Sometimes it contain like several types. It's like a community. If you have seen some paper, they're talking about the microbial community of plant that stay healthy and the plant that stay unhealthy. They found that the microbial community are different, right? Healthy plant and unhealthy plant. So it means that the community of the PGPR or bacteria around the rhizosphere is very affected, right? It can affect the plant health. And it's not just only one single bacteria. You have to use a community or a complex. And this is another way that we have to do the multiplex inoculum or engineer the microbiome using the PGPR. Yes. So this is for the 21st century skill, how to make a food security. Actually, for me, I do not against the crop system using the chemical compound or chemical fertilizer. I do not against it if we use it in the correct way. If we use it in the right amount. Normally, in Thailand, I show in my case, Thai farmer, they do not analyze their soils before using fertilizer. So they just keep using chemical fertilizer a lot, heavy inoculation, like 16 all the time. So in this case, once you apply too much chemical fertilizer, the soils become bad, right? Because the soil structure, the soil, the organic matter is reduced. So in this case, if you apply too much, it makes the bad effect on plant growth. But if you put in the good amount, in the appropriate amount, this is the good way to promote plant growth. Because when I talk about the PGPR can produce nutrient available, but to fix nitrogen is very little amount compared to if you put the urea, right? PGPR is just a little amount. Or if you use the PGPR that can have the phosphate or potassium solubilizer, it just can help to solubilize, but you need to put the source of phosphate into the soil. This bacteria cannot make phosphorus or potassium by itself, but it just solubilize the source of phosphate that precipitate in the soil. This one can just to help, right? So to me, biofertilizer, except rhizobium with legum, this one can use instead of chemical fertilizer. But to me, the PGPR can be used as a assistant. Okay, I can say it's the assistant. You can use chemical fertilizer and PGPR together. This one can promote the plant growth better than using PGPR alone, or you can use PGPR with the compost or organic fertilizer. Okay, so when you use with the chemical fertilizer, less amount of chemical fertilizer is needed, because if you use too much, the high EC of the chemical fertilizer will affect the death of the microorganism. Okay, so in conclusion, for the security of food security, you can use chemical fertilizer, you can use PGPR, or you can use organic fertilizer. But the way of application, you need to understand it and use it in the correct way and safety. Yes.
Okay, so the last question for today from Aurora.
Okay, for Aurora, I want to ask about the mechanism of the bacteria to produce some volatile compounds, especially in gas form to support plant growth. Are there any specific factor, maybe genes or certain conditions, which can induce production of volatile compound or bacteria? Okay, this one I would say that I do not have much knowledge in this one. I just found this one in the paper, but I did not study on this mechanism. But yes, to me, I think it should have some specific gene that produce this compound, and it must have a specific condition to induce the compound production or gas production. Yes, but I'm sorry that I cannot tell you about the deep mechanism on this one. But yes, there are many factors. Okay. All right. Thank you very much.
Okay, I think that's all for all the question from I think most of them from the student and also from the lecturer as well. So again, thank you very much, Ajahn Op, for your interesting and remarkable presentation. I think we all have my student will have more insight and also new knowledge regarding this plant microbe interaction. And I also hope that later we can collaborate to do kind of research together, because you know, Thailand and Indonesia has diversity in microbe very much. So then we can like exploit in the good way for the farmers and in the agricultural field. So again, thank you very much for your presentation and for your time, for your effort to give us new insight on this plant microbe interaction. And on behalf of Biotechnology Study Program, we say thank you very much again. Okay.
Thank you very much for Ajahn Anto to invite me. It's my pleasure.
Thank you very much.
Yes, thank you very much. Okay. So thank you very much. And if students have more questions, yes, please contact me.
Okay.
I'll give them my email.
Okay, cool.
Okay, cool. Thank you again.
Thank you. Bye-bye.
Bye.